Hello students and welcome to the online lecture series. Myself Dr. S.R. Patil. I am working as an assistant professor at the Department of Chemistry in the Maratha Vidya Prasarak Samajas S.VK.T. Arts, Commerce and Science College, Deolali camp. Dear student, once again this session for the TYBSc student and the subject name is the Introduction to Medicinal Chemistry. Dear student, we know that we had started the first chapter from this course, Introduction to Medicinal Chemistry, and the name of chapter is An Introduction to Drugs, Their Action and Immunobiologicals. In the first session, we had discussed about the introduction about the drug molecule, then we had discussed need of new drug molecule. After that, in the last session, we had discussed about the term drug resistance. Then we had also discussed how to improve in properties of existing drugs. After that, we had discussed in the last session identification of new disease, how to identify the new disease and what is the importance of identification of new disease during the process of drug designing and drug development. Along with that, we had also discussed some historical background of the drug discovery and drug design in our previous sessions. Now, today we discuss about the next point. We discuss the next point from the same chapter, that is the sources of drugs. In that, we had discussed different sources of drugs. First, we will discuss about the plant sources. Then, next source for the drug is the animal source. Next one is the microbial source. Another important source for the drug molecule is the marine sources. Along with that, some mineral sources, some semi-synthetic sources and synthetic sources. All these are the sources of drugs, and today's session we have discussed all these sources one by one with the examples. Now, what is the learning objective of today's session? After the today's session, students are able to learn about the basic of medicinal chemistry. In that, different terms are included, and these terms are new for the TYBSc student because this course is introduced first time at the TYBSc level, okay, medicinal chemistry. So, another important point cleared after this lecture is the biophysical properties and overview of the basic concept of the traditional systems of the medicines. These are the different terms including in the medicinal chemistry, and all these terms and some basic background of medicinal chemistry we learn in this session. Along with these basic terms or basic background of medicinal chemistry, we will also learn to the overview of overall process of the drug discovery. Okay. After that, students are able to learn about the role played by the medicinal chemistry in this process of drug designing and drug development. These are some learning objectives of today's session. Now, we start with the main point of today's session is the sources of drugs. We know that as the earlier discussion in the previous session, different natural products from the animals, vegetable and mineral sources were the main sources of drug. The drug molecules are obtained from these different type of main sources, and the drugs were discovered mostly through continuous experiments and the evaluation process. We know that because we are the chemist and we know that how to different steps employed for the discovery and the process of the drug designing. In that, different sources, out of different sources, first we discuss about the plant sources, the drug obtained from the plant sources. We know that the plants have been the drug sources for the centuries, and the different parts of the plant are used as a drugs. And in this part, we include the roots, seeds, fruits, stem, barks, and leaves also. So these are the different parts from which we can isolate the drug molecule, or we can use directly this part as a drug. If these parts are directly used without any processing, then such drugs are called as a crude drugs. Okay, and when we use this part after the processing, okay, doing some process, then these type of drugs are called as a prepared drugs. Remember that when we use these plant parts without any processing, such a type of drug is called as a crude drugs. And when we proceed on that parts and then we use as a drug, it is called as a prepared drugs. So these different parts of plant have medicinal properties, okay, because of the presence of many biological active compounds. Many biological active compounds are present in the different parts of the plants. So most of these compounds have already been isolated and identified in a literature. All these information are available. In that, for example, we discuss or we take in consideration anti-malarial drug like a quinone, okay, which is isolated or identified from the cinchona bark. Another example from the drugs from isolated from these different parts of the plant, in that the cardiac stimulant digitals from the foxgloves and the antidepressant rasparin isolated from the Rolfia's serpentina. Okay, all these different type of drugs which are compounds which are already isolated and identified from the different parts of the plant. We can take another example of a such type of compound is the paclitaxels, and it is the potential anti-cancer drugs which is isolated from the Pacific oak tree. Okay, then all these are some successful examples of a drug obtained from the different plant sources. This is the structures of quinone and rasparins from the isolated from the plant sources. Next drug source is the animal source. Okay, we know that the medical substances derived from the animal body or animal secretions. Fluids or glands are called as the drug source from the animals. Okay, once again I repeat different animal sources. The drug substances or medicinal important substances are isolated or derived from the animal bodies or animal secretions, fluids or glands. Okay, and such a type of drugs are the source from the different animals. For example, the cod liver oil from the CADD saps, then insulin from the bovines or the porcines pancreas, then hiridine which is obtained from the European medical leech, and heparin is the one of the example from the Mexican medical leech, are some example of compounds or drugs which is obtained from the animal sources. Another important source of the drug is the microbial source. We know that the large number of life-saving drugs have been derived from the microorganisms, and the random screening of a soil sample leads to the discovery of the streptomycin and the tetracycline antibiotics. These are some examples of the microbial sources, streptomycin. And these antibiotics inspired the development of many other drugs from their groups. Okay, for example, penicillin produced by Penicillium chrysogenium, then streptomycin from the Streptomycin graecis. Then another example is the chloroampheniscols from the Streptomycins venezuela and neomycin from the Streptomycin fredis. Okay. These are the some example of compound which is obtained from the microbial sources, and the structure of streptomycins and the tetracyclines are drugs which is obtained from the microbial sources. Next important source of the drug molecule is the marine source. Okay. Many biologically active compounds have been identified and isolated from the marine sources, in that from the fish, corals, sponges and the marine microorganisms for the treatment of many different diseases. Okay. For example, the cuarsins A from a marine Cyanobacterium lingbytes majuscules is the strong cytotoxic agent. Okay. This is the structure of cuarsin A which is obtained from the marine source. Then further, next source is a mineral source. Okay. The next source for the drugs are the important source are the mineral sources. In that the metallic and non-metallic minerals are also known for their therapeutic activities. For example, the ferrous sulfate for iron deficiency, which is used for the iron deficiency. Then another marine source, magnesium sulfate, is used for the constipation. Then further example of the marine source is the mineral source is the aluminum hydroxide and the sodium bicarbonate for the acidity and peptic ulcer are used in various formulations. These are the mineral sources which are used for making the drugs formulation, various formulation of drugs. Okay. For example, the aluminum hydroxide and sodium bicarbonate, which is used for the acidity and the peptic ulcer, and which is used in various formulation of the drugs. Another important source of the drug molecule is the semisynthetic source. We know that such a type of drugs are obtained on the chemical modifications of naturally occurring substances to improving their potency, efficacy and regulate side effect. These are the semisynthetic source. Okay. Some naturally occurring compounds are taken into consideration and chemically improved, okay, their properties like the potency, efficacy and regulate side effect, and used as a semisynthetic drugs. Also, the basic skeleton is intact during the chemical alterations of that naturally occurring compounds. And in that semisynthetic sources, we consider the different type of examples like heroines from the morphine, then bromoscopolamines from bromoscopolamines from the scopolamines, and the ampicillin from the penicillin. This is a well famous drug. We know that ampicillin, which is prepared from the penicillin. Okay, this is the structure of ampicillin, which is a semisynthetic drug, or which is obtained from the natural source by improving some properties, or by chemical alteration improve their properties. This is included in the semisynthetic source. Next important source of the drug molecule is the synthetic source. We know that purely synthetic drugs are the outcomes of a modern approach of drug development, okay, and which involves the study of pathology of the disease to find point of interventions and suggest the possible lead compounds. Okay. In the synthetic sources, different type of precursor or starting materials or we can say that the lead compound, starting compound, which are synthesized in the laboratory by the different chemists, different pharmacists, or different medical professionals, experts are worked in this area to identify or to synthesize the new drug molecule by the synthetic route. We know that what is meant by lead compound. Already we discussed this term in our previous session. Okay, and these compounds are synthesized and eventually from the pharmacological action. These compounds are synthesized in laboratory by using different type of chemicals and evaluated for their pharmacological actions in different type of laboratories. They check their biological activity, and after that they are used as a different drug molecules. This is one of the example of semisynthetic drugs, that is the axanoxin, which is synthesized in the laboratory. We know that most suitable lead compound is then used to make the structural analogs to make a new compound. Different type of analogs are synthesized in the laboratory. Okay, there are different natural products, natural drugs are available. They are showing the different type of biological activities, and the analogs of such a type of biologically active compounds are synthesized in the laboratory. Okay, or the lead compound are synthesized in the laboratory. Okay, and in that the structural similarities are there, okay, which are suitable for clinical use in terms of their biological action and toxicity also. When the synthetic methods are used for the preparation of such a type of lead compound and its analogs, it must be kept in mind that the reaction should give higher yield in order to make the process commercially viable. We know that in the first session we discussed about the pharmaceutical sector, how big it is. Pharmaceutical sector is how much investment are made in this pharmaceutical sector. So it is important to concentrate on the commercial value of all these processes. So during the synthetic process of modification or the synthesis of a new drug molecule or synthesis of a new lead compound and their analogs, it most important to improve the higher yield, and due to the higher yield the commercialization of that process increases, or it may be valuable, because all these processes are very more and maximum money or very costly. So all these processes very costly. So therefore it is take in mind to consider the commercial value of all these processes. So if the synthesis requires a large number of steps, then the percentage yield of every step becomes very important. We know that different type of for the synthesis of different type of molecules, different type of lead compounds, there are number of steps are required to perform in the laboratory. That is the multi-step synthesis, we call it as a multi-step synthesis, and all these work are done in the research and development laboratories from the different type of pharmaceutical industries. And when we think about the multi-step synthesis of a new lead compound or any new biologically active compound in the laboratory, so we have to take in a consideration about the percentage yield of each and every steps, okay, because we take the compound from first step for the second step. So it is most important the percentage yield of every step from the multi-step synthesis, and then it is also important the identification, the isolation, purification of products in each and every step, because in some step the isolation is difficult, and in such a time the yield is less or yield is reduced. When yield is reduced, then it is very difficult to perform the next step. Therefore, the isolation, identification, isolation, and the purification also, purification of that product is most important. When any product is impure from any step, any one step, then next step is not possible, or that impurity is carried forward on the next step, and therefore purification of that product is most important. So during the multi-step synthesis of any lead compound or any biologically active compound in the laboratory, different type of point of view is under consideration, in that the percentage yield, identification, isolation, and the purification of product. Then the final product should be easy for a successful synthetic approach. This is the most important point as concerned with the multi-step synthesis. If the reaction can give rise to stereoisomers, okay, then the synthesis of desired isomer in a higher yield becomes a challenge in front of the synthetic chemist, and reaction needs to be modified to make it stereospecific. Okay, all these steps should be easy to carry out at a large scale for the industrial production of the molecules. Okay, this is a very important point as concerned with the multi-step synthesis. During the process of discovery and design of the new drug molecule or new lead compound, it is a must be considered the stereochemistry of that molecule. Example of such a type of synthetic source, synthetic drug, is the ozenoxin. Okay, is a purely synthetic drug which is a design for the treatment of a skin infection. Okay, in that it is a very effective against the wide range of gram-positive and negative bacteria. For example, aureus and epidermidis and the propionobacterium ascens. These are gram-positive and gram-negative bacteria, and the drug, synthetic drug, is very effective against all these bacteria. So the drug initiate cell death by binding to DNA topoisomerase 4 and the DNA gyrase. In recent time, computers plays the major role in the screening and designing of new compound, that is called as a computer aided drug designing and development. Okay, so this is a CADD, computer aided drug designing and discovery, CADD. In this process, takes into account the available data of various properties of a large number of drugs and drug-like structures, and the predict new structures for a given physiological conditions. And in these computer aided drug discovery and design, okay, be carried out with one software. These different type of softwares are used for the CADD. Okay, this process of CADD cannot be carried out with one software. They require many softwares. Okay, a very large number of different type of applications have been developed to cover all the aspect of the drug designing in the CADD. Okay, with the help of CADD, in that covering all the aspects and their utility cannot be converted here because the quantums of the information is too large, and hence some of the tools are mentioned here in that next table. Okay, and in order to give us some idea of the total concept of CADD, computer aided drug designing and discovery. As I earlier said that number of softwares are used in the CADD process. There is no any one software, number of softwares are there. Okay, and this table shows the software used in a CADD. In the first column, there is a type of parameter, okay, which is studied by the software. In the second column, the name of software, and in the third column, there is a detail of predictions or the calculations done by the software. The first parameter is a pharmacokinetics. Okay, and this pharmacokinetic parameter is studied by the software DDD plus, 3 times D, DDD plus. And from this software, we can predict or calculate the study of dissolution and disintegration of drugs. Study of dissolution and disintegration of drugs. Next type of parameter is the ligand protein interaction, and this interaction is studied by the software name is Autodoc. Okay, and we can predict from that study, we can predict the binding interaction of drug with the target protein, ligand protein interaction, binding interaction by the help of Autodoc software. Another important parameter is the structural or structure activity relationship. Okay, the structure activity relationship is studied by using the software PASS. Okay, and from that we can predict about the develop model of the structure activity relationship of a large number of compounds. So another important parameter of the drug compound is the image development analysis, and this image development analysis is studied by using the software PMOLS and the Discovery Studio. Okay, PMOLS and Discovery Studio, this is the name of the software, and from that software we can predict or calculate the viewings and the analyzing protein data and the development image of the protein ligand interaction. Okay. Another important parameter studied by the software is the genome analysis, and for the genome analysis study the software used is a BioSuite. Okay, BioSuite, and from that study we can say that the genes sequencing to find the abnormalities. We can study of a genome sequencing to find out the abnormalities. Another important parameter of drug molecule is the bioinformatics, and these bioinformatic parameters of the drug molecule is or any compound, biologically active compound, is studied by using the software REST, R-E-S-T. Okay, and from that we analysis of the change in a genes expressions crossing points, that is a CP of a amplification curve. This is the prediction or calculation done by the REST software for the parameter bioinformatics. Okay, these are the different software which are used for the computer aided drug designing and discovery. Okay, and in that different parameters are studied, and by studying that parameter we calculate or we predict a different type of mechanism which are done or activity which is shown by that drug molecule. Okay, these are some softwares which are used in a CADD process, computer aided discovery and development process. So dear student, at the last of today's session, quickly recap on the summary of today's session. In today's session we had discussed about the sources of drugs. Okay, in that first source we have discussed is a plant source. Then another source of drug we had discussed is the animal sources. After that we had discussed mineral sources for the drug. Next point we had discussed in today's session is the marine sources, then mineral sources, then some semi-synthetic sources, and final source we had discussed in today's session is a synthetic source. All these sources of drug molecule which we had discussed in the today's session. This is session assignment for the students. Okay, there is only one question, but it is a broad question. Okay, and the question is write note on drug sources. In that we have to write down the all these sources with example. Okay, and this is a broad question, long answer question. So write down this answer of this question and send on the Google Classroom for the TYBSc class. This is all about the today's session. Now in the next session we will discuss on the next point, that is the classification of drugs. Generally drugs are classified in three different classes. First class, it is classified by their pharmacological effects. Second class, the drugs are classified by their chemical structures, and the third class, drugs are classified by their target system. All these points of the drug classification we will discuss in our next session. Okay, thank you.